That's why I went into
There are a lot of people that were like, well, I don't know. Professor Rob is just well, I had 3 tests. It doesn't matter. Like as somebody wanted to like in the past, somebody I'm aware. I honestly I got a lot of like half I couldn't tell if it's X or the same. I got this. There's never a point where I'm not yeah, I got this.
Yeah. And then they just like showed up. And then I'm gonna ask you yeah. Which was the whole 500
I got better. Precisely, I know. Like, I know the incantation.
It is her though.
He actually looked just, I think, took the because a lot of things. I like how he was like it's like that. I don't know what that's here. Oh, but the
Let's see.
That's what I have different favorite figures, a different color.
this is like I know.
Yeah, I'll show you what I have. I think
You got your haircut. Okay, so I should first ask, are there any questions pertaining to the homework that I can answer? This is your time. Yes, Alex.
I'm confused for a little bit with the second problem.
Okay.
Are we trying to calculate the error for any combination of x, y, z that there would be an error at all? So that either C is wrong, S is wrong, or C and S are wrong?
Correct. So did you all get my email from yesterday?
Yeah.
Yeah. So that didn't clear this up?
Not
Okay.
for me. So
Okay.
it's still just one number we want to find for each one then?
Yeah. So let me try to explain again. Okay, this isn't the exact circuit, but I think it's close. So this is carry and sum, or the other way around, with x, y, x, z. I'm getting it wrong here, but the essential point is that there's 5 gates and 2 outputs. And so Alex's question is a good one. It's also somebody else raised this in an email. I want you to compute the probability that there's an error in the output. So probability of the error in the output. Now, there are 2 outputs, C and S. So what does that mean? So I'm pessimistic here, and let's consider the circuit to be an error if either C or S are an error. So if we're supposed to get, let's come up with an example here. Suppose we have 0, 0, 0, 0. That'll give us 0, 0, 0, 0, 0. Put another one there. So we're supposed to get 0 and 0 here. But suppose that this gate has an error. That means that instead we'll get a 1. This is a case where the entire circuit is an error, so that contributes to the probability of error. Does that answer the question?
Yeah.
Yeah. Okay. Any further questions on this one or anyone else? Yeah.
So I guess would you like us to enumerate over all the possibilities of gates failing
Yeah.
for every single combination of X, Y, and Z?
Correct.
Okay.
So there's 8 combinations of X, Y, and Z, and there's 5 gates, so there's 32 possibilities per combination. Some of those, of those 32 possibilities, one of them is that all the gates are correct. So obviously the answer is going to be correct for that one. Of the other 31 possible cases, in some cases you'll get an error, some cases you won't. So only take those cases where you will get an error and sum up those cases. So definitely on the tedious side, because that's 256 cases, but if you're doing this by hand, you can simplify this quite a bit by noticing some symmetries. Like, for instance, the circuit is symmetric with respect to x and y. So if you interchange x and y, you get exactly the same circuit. So that immediately cuts the number of possibilities in half. And you could do some further simplifications there. Does that make sense?
Yeah.
Okay. Any other questions on 2 or any other problem? Yes?
Can we calculate the probability of failure for one gate, one output once, and then the other output in the next step, and then sum them together because it's arithmetic, right?
They're independent. Well, are they independent?
Yeah, because the output to this gate is the one by the input, this independent input. Yeah, this one, and the other one. No, not that one. The other input to that. Yes, yes.
Oh, okay. Yes, these 2.
So
Yeah.
can we not calculate the failure of the upper one separately, and then for the lower one using these gates only, right?
Uh-huh.
The gates in the bottom, and then can we not sum those up?
I'm still not quite following. So sum up
Calculate the individual probabilities of failure.
for each gate?
Yes.
But
No,
that's
for each output.
for each output.
Yes, correct.
For C versus S. Okay. Still not quite following here. C versus S. So compute the probability that C fails for a given input combination. Compute the probability that S fails, compute the probability that C fails, and then sum them up. But the OR is not a sum of the probabilities. So A, the probability of A happening or the probability of B happening is not the probability of A happening plus the probability of B happening.
Yeah.
Right?
You can union.
So you have to take the probability of A failing plus the probability of B failing minus the probability of both of them happening. But then you may as well look at each case and take the logical OR. Does that make sense? But I think you're understanding exactly what's needed here. Okay. Anything else? Is everybody clear on the percolation problem? I tried to clarify what to do. In the problem specification, I didn't include specific numbers, which is always a mistake. My intent for that problem, and in fact for all these problems, is not to look to see if you guys get a precise numerical answer, and if you're off by 1 decimal point, put a big X. Obviously, the intent here is to get you thinking about these problems, and for some of them to appreciate that there's something interesting behind it. So I figured when writing the problem, all right, just leave it flexible. Let the students explore percolation for these two different models and see if they can demonstrate the S-shaped curve occurring. And my intent, if I was grading it, would be that's what I'm looking for. But that leaves it open, and leaving it open is never helpful for students. So what I should have done and what I'll do in the future is always say, here's the general problem. Here's what I'm looking for in a solution. Evaluate it for these specific values. So you can choose a specific value of n and a specific value of m and show the results for them. Yeah, John?
I do have a question. If we did it bidirectionally and using different values
Yeah.
than you did before you sent the emails, is it okay to
It
do
is.
that?
It is. So that's always going to be, if ever on an email, I provide clarification that's actually somewhat changing the specification of the problem. You can always do it the original way. So if you wanted to have bidirectional edges for part 4A, that's fine. Just, of course, kind of emphasize it in your write-up. Say, I assume bidirectional edges. Yeah. Okay. Similarly, if someone else answered this problem where they didn't choose my values of n and m or r, that's fine too. You can just hand it in that way. Okay. All right. No further questions? Good. So let's pick up where we left off last time, which is a discussion about how a set of chemical reactions can compute probabilistically. Now recall that when we discussed chemical kinetics, we did so in two different ways, either using deterministic differential equations or using probabilistic discrete events. And so according to the model of probabilistic discrete events, chemistry is inherently probabilistic. And as I think I've emphasized before, this is, by some measures, a very accurate way to view reality, or to view, what's the word for it, I guess, chemistry, the way molecules interact. If you have molecules of a gas in a cylinder, we're not getting here into the level of quantum mechanics and that kind of randomness, but at this level of interpretation, where the molecules are going and which molecules collide with which molecules is random. That's an accurate way to view what's going on. The amount of bouncing around that occurs is a function of the concentrations and the temperature, but there's randomness there. And so, in a sense, it's not surprising that we should say we want to design a set of chemical reactions that computes probabilistically. But this is, in fact, a controversial view. It's a controversial view because, well, to give some context, I don't know if I should launch into this here or in a moment, since I've begun talking about it. So we have a system of some kind. Let's say you're trying to model the biochemistry inside a cell, or you're trying to model a particular set of chemical reactions that you're studying in a test tube. You can have inputs, X and Y, and obviously the types of molecules and the concentrations you supply will dictate what happens. But you can imagine a situation where you put in quantities of X and Y, and because the underlying reactions are random, you do the experiment once, and you get some output value of Z. Let's say Z is greater than 100. You do the experiment again, and this time you get Z is less than 50. So that, to me, without any further discussion, doesn't seem like an outrageous claim. And yet this is controversial, because people who have experience with chemistry will say, well, I don't know of any situations where you supply the exact same setup, the exact same concentration of the reactants, and you do the experiment once, one thing happens, and you do the experiment another time, something else happens. So, Jesse, what's your view on this? Like, do you know of such a system where, very simple system, everything is the same, you do the experiment once, something happens, do the experiment, something else, another time something else happens?
Done properly, that shouldn't happen.
But exactly, so that's your view. But if done properly, right. And so that's exactly what I'm going to go into. That's the view that people have. But a priori, if what's going on underneath, if we accept that randomness is a good way to describe what's going on, why wouldn't you expect this to happen?
I mean, in all the randomness, there is a certain amount of order and construction, and as you said, these chemical reactions are sort of like rules that
Yes.
the system follows, and because of that, certain outcomes are far more likely than others.
So, yeah, that's a very good answer, very articulate. I think what you're saying is that the law of large numbers kind of kicks in here. That you may have randomness, but by the law of large numbers, things, you presumably got lots of such molecules, the same thing tends to happen, even if things happen individually. And I think that's the common experience of people who do experiments like this, and so it becomes entrenched in their view. Well, what I'm going to suggest today is an explicit way to try to set up this kind of occurrence. But first, just a little bit more about the controversy here. So Jesse's initial reaction was, well, if you do the exact same experiment twice and you get different outcomes, then perhaps you haven't been careful enough. And again, this is a common view. So the label that's given among the community that debates these issues is they call it intrinsic versus extrinsic noise. So intrinsic versus extrinsic. These are, as with so many labels, dumb labels, because unless you know exactly what they mean, they don't help. But what the community means is extrinsic noise. Noise is the key word here. Extrinsic noise is simply noise that is there, and by noise what they mean is variations in the setup. The noise is there, you just don't know about it if you perform the experiment and you get different results. So you think you've set it up so the conditions are absolutely identical in trial 1 versus trial 2. But in fact, there are differences. Those differences could be anything that you imagine, but it could be slight differences in temperature, it could be slight differences in the chemical composition of what you provide. In the case of cells, you can imagine that no two cells are identical. So there's all of that. And so this view is that when you observe what appears to be random outcomes from an experiment, this is because you haven't accounted for, and perhaps you can't account for because it's too complex, all the sources of variation that could be present in your experimental setup. That's extrinsic noise. And that kind of conforms to the view that you don't get randomness from chemical reactions. But intrinsic noise is the opposite, and that's the one which we're interested in, which is that there is inherent randomness in what goes on, and that inherent randomness, which is not due to your ignorance, but is just due to the physics of what's going on, that accounts for the probabilistic outcome. And interesting or not interesting, papers go back and forth where a researcher will publish an example of intrinsic noise that he claims. And I'll show you a fascinating one in a moment. It's called the lambda phage, which I've spoken about. And people will be interested in that, and then a whole sequence of papers will come up afterwards saying, no, 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 person was wrong. Everything he saw experimentally was actually due to extrinsic noise. And that pattern has repeated. And interesting or not, well, I'd like your take on what I'll present now, which is what I think is just a simple, straightforward scheme. I've never done it with chemistry of any kind, but a simple, straightforward scheme that should unequivocally give us probabilistic outcomes which are based upon the true randomness of the system, or intrinsic noise. Okay, so let me explain that. It won't take too long, and then I'll return to the issue of lambda phage and an actual example where this occurs in practice. So the setup here is the one I've set up, I think, twice already, which is what we're aiming for is going to be a set of chemical reactions that gives us a probabilistic response. The motivation for it could be to settle the debate of whether there exists intrinsic noise or not. But it could also be a much more practical one, which is, and this is my particular take here, that if we could achieve a probabilistic response from a set of chemical reactions that are engineered into a bacterium, then this could be a powerful mechanism. It could be a powerful mechanism because probability is a very different dial that we can turn compared to other dials. So imagine each bacterium is designed to produce a specific compound. That compound would be something useful, like a compound to kill cancerous drugs. But in order to achieve a very sophisticated amount of control, you might say, well, what we want is a knob in which we can carefully control the amount of the compound that each bacterium produces. But that knob might be very, very hard to control, simply because each little device, the device here being a bacterium, I'm using some jargon, each little device is very limited in the specific levels that we can control. A typical example is you can have the chemistry within a bacterium produce a little puff of green, and that's it. You can't control to within 5 decimal places the exact concentration of the puff of green that it produces. You can basically set it up so that it produces a little puff of green, which is just an amount that saturates quickly, or it doesn't. So it means that turning the dial of how big the puff is, that's very hard to turn, that knob. And the other knob that's hard to turn is it's hard to control. So imagine now, I'm using all kinds of metaphors, but imagine that these bacteria are soldiers on the battlefield. It's very hard to set up different skill sets for the soldiers. So you can't declare these guys to be infantry, those guys to be cavalry. In other words, to have the bacteria do different functions or perhaps respond differently. That's also hard to do, because the bacteria are going to be identical. Each one is created from the same blueprint. And it can be hard to just achieve that kind of control over your army. So my argument here, and as with all such things in this class, I should be careful not to present it as an accepted view, because this is a view that I'm suggesting. So I'd appreciate opinions on it. But the view that I'm suggesting is it could be very useful instead to have your knob be probability. So to have your knob, I've gone through this a few times. Your knob be the probability of whether a given bacteria either produces a puff or doesn't produce a puff. If you can control that, then you can control the overall quantity. Does everyone buy this? Okay. So the question is, how can we do it? So I think I stepped through this last time, that we may want to produce or not produce with a certain probability. That's one goal. Another goal is, or a more specific goal, might be to produce or not produce not just one type, but a range of types. So produce a green puff, blue puff, red puff. These can be mutually exclusive events. Either produce green, or produce blue, or produce red, with specific probabilities. Or we could have logical combinations, so getting more sophisticated. Produce green puff and blue puff, or produce blue puff and red puff. But in each case, we're not trying to control the amount of green or the amount of blue. It's either a puff or no puff. And the further generalization, and the important one here, is that we don't want these to be fixed, the probabilities. Fixed probabilities could still be pretty useful. Suppose the constraints are that you want to design your system to deliver a specific amount, but you're limited in how you can control whether that amount is delivered. You know how much you want in advance, but you're limited in how you can control it. It could be very useful to just have the probabilistic response. But even better would be to set it up so that the probability of it happening or not happening is dependent upon an input that you supply. Now, each bacterium sees the same input. So each sees the same input, so you're controlling all of them simultaneously with the same input. You're not selectively deciding or selectively changing the probabilities. But if you can globally control the probabilities, you're globally controlling the amount. So that all sounds great, I hope. But how do we do it? So let's try to wake up the audience here a little bit. How about Alex? How can we do this?
How can we make the fractional response?
Yeah. So a fractional response, so I think what I'll present is going to be simple. It's going to be concentrations of x and y in. And I want the probability of something happening or not happening to be what the result is. So I supply x and y, and it's going to be the relative amounts of x and y that matter. In particular, the relative amounts, we're going to go for x over x plus y. If I supply 30 of x and 70 of y, then I'm going to have 0.3 as this value, and 0.3 is going to be the probability of getting green. But this is non-trivial, I think, and conceptually interesting, because we're going from quantities, which are deterministic, we supply them, to probabilities. We want to do that with chemical reactions. So any ideas at all? Your name is Ron, right?
Yeah. I guess when we were going over previously, we had like a list of chemical reactions.
Yeah.
And we were able to calculate what probability each reaction fired with.
Correct.
So you could, I guess, mess around with the sort of chemical
Very,
pathway
very good.
in order to get specific probabilities that you want.
Right. So this is exactly what I'm trying to prod you, not to come up with a final solution. But as Rowan said, previously we described a set of chemical reactions as simply a set of rules, with the probability of each rule firing being something that we can calculate. And so, and that calculation is based upon the quantities present. So it seems like we have all the ingredients. We go from quantities to the probabilities of things happening. But that's just one set of probabilities, which is the initial set of probabilities. Once that rule fires, the quantities change, the probabilities change. And the system has a life of its own. And here what we want is just a very clear, simple design paradigm to achieve an overall probability of outcome as a function of the quantities we supply. So I'm just trying to prod you guys. So,
a huge amount of the quantity?
we could. Sure. But how will huge help us here?
Because it will take a longer time for the ratios to change.
Mm-hmm.
So we'll have almost the similar concentrations, like relative concentrations for a longer period of time.
Uh-huh. But think of the goal. So the goal that I want is going to be produce green or don't produce green. So we can start off with huge quantities or small quantities. And then I want the system to fire, 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 fire. And if I supply x over x plus y with, let's say, 30 over 30 plus 70 being the values, if I do this, I want 0.3 of the time to get a puff of green. 0.7 of the time, I don't want a puff of green. And this, how can we, and so we can set up reactions that produce some amount of green, but I want the amount of green that it produces just to be fixed, but with a certain probability. Yeah, Ron?
Because one way you can do it is like encoded in their DNA that they just like produce something and then die. Then you can like continuously put more bacteria.
Yeah.
That way the probability stays constant.
Uh-huh. But here, everything, and that's a good suggestion, but everything that I'm presenting is level of chemical reactions. So we're not talking about, the level of the cell and death and how to program it. Yeah.
maybe the X and Y, they are like catalysts which are controlling which side reaction goes, because like in normal combustion, you have like C plus O2, it
Yeah.
can go to CO2.
Yeah.
But if you have less O, it can go to CO.
Yeah, that's a very good idea. So in essence, you can view X and Y, they will be catalysts of a sort. Actually, I'm going to introduce a few reactions, and some of the reactions are going to be exactly of the catalytic type, where if we have X, we start to produce a puff of green, and the more green we have, so X doesn't get consumed. It simply facilitates the reaction and makes green. So the more X we have, the more green we have. And we can do the same for Y, but then, how do we bias it so that either one outcome happens or the other outcome, and it happens with the probability that we control? But these are very good ideas. So any other ideas before I begin to explain it? Yes, your name is Bishwesh. Yeah.
For at least whether it will be produced or not, you could have two mutually inhibiting reactions.
Very good.
So it's almost like a switch.
Yeah.
Whether it will be in one state or the next.
Very good. So these are the two ingredients. One is the catalytic ingredient, which is once something gets going, we want it to really get going. The other ingredient is once something gets going, you want it to inhibit the other outcome. And that's basically all we need. So let me try to explain. So for the sake of explanation, I'm going to go to just a specific design criteria. So suppose that, I hope I don't mess up the alphabetic letters that I use for the different types here. So we're going to have types D1, D2, and D3. And what we want is a response that's going to be, so this is a specific design here, a response that's going to be, let's say, 0.3, 0.4, and 0.3. In a moment, I'll show you how those values are programmable. But it means that we either want D1, or we want D2, or we want D3. And we want those with specific probabilities. So think of puffs, mutually exclusive puffs of the different types. So to do this, I'm going to introduce this as a sequence of reactions. And I think, helpful or not, I think I introduced 3 reactions per slide. But the full set of reactions is going to be something like 12 or 15. So bear with me here. But the paradigm is I'm going to set up initializing reactions. And these are very boring-looking reactions. I just have types E1, E2, and E3. And these each have the same rate constant 1. And they produce different types, D1, D2, or D3. But everything is symmetric. And so all things being equal, if you just had this set of reactions and you supplied some ratio of quantities E1, E2, and E3, what can you say about the system? So Jason, what can you say about the system? So suppose that I give you 30 of E1, 50 of E2, and 20 of E3.
The final ratios of D1, D2, D3 will be the same as that of E1, E2, E3.
Probably. But will it? Yeah. But here, I guess what I'm going for is what can we say if we're running a simulation? What can we say right at the outset? So nothing has started yet. I dump E1, E2, and E3 into a test tube. Then something's going to happen. What can you say about that first thing that happens?
E2 might happen first.
It might happen first, but the probability that E2 happens first is exactly E2 over the sum of E1 plus E2 and E3. So this is everything symmetric here. The probability of the first thing happening is simply dictated by the ratios. Now that in itself doesn't say anything interesting because as we were discussing before, that's the first thing happening, but many things are going to happen. Perhaps millions of reactions are going to happen, and so it's not clear yet that anything interesting has happened. But it's in this step that we actually program the response. And this, I'd appreciate opinions on it, because here we're really going at the heart of the intrinsic noise. I'm claiming that you pour E1, E2, and E3 into the system, And something has to happen first. And what happens first will have a probabilistic outcome based upon what I show you next. So can anyone guess what comes next? So your name was? Rishad. So the catalytic aspect of it, what would you suggest comes next in terms of the catalytic aspect?
Before continuing,
Yeah.
why are you considering this as non-parallel reactions? I mean, why do you say E2 will happen first?
Oh, but they are parallel.
Right.
Well, and this is going to the heart of it. It'll make sense in a bit, I think. Because you're saying, well, E1, E2, and E3, we can have a bunch of those happening in parallel. But you'll see in a moment why that's unlikely to happen. Okay, so to set up the metaphor here, maybe this will help you describe where I'm going. I haven't introduced this metaphor so far. Let's see if it works. Imagine you have a bunch of soldiers that are lined up, E1 and E2 and E3. And they're all just lined up motionless. Randomly, one of the soldiers takes a step forward. That's going to be the first reaction. And if nothing else was set up, then the other soldiers would probably take a step forward too. But suppose that first soldier who steps forward, what does he do? He turns around and he shoots all his friends. Well, then the reactions won't happen in parallel. The first soldier has killed all of his enemies, or all of his friends, whatever. And so now he's the only soldier that's alive. So the reactions won't happen in parallel. That's the mutual inhibition part that Bishwesh described. The catalytic part is suppose that once you step forward, what's an analogy here? You start having babies, and you make many of yourself. Sorry, it's hard to get the metaphors right here. So that's the catalytic part. So how can we set up the metaphor where the soldier kills all his friends and has makes lots of copies of himself? If we can achieve that, then we won't have the reactions happening in parallel. We'll have a winner. And if we have a winner, then only one thing will have happened, in the sense that we'll have one quantity that's produced. So, ideas?
So the products block further reactions.
Yeah?
Products inhibit other reactions.
Right. Okay. So let me try to set this up.
So one way I can take it off is like we have adsorption, like in hydrogen adsorption, a molecule comes to a catalyst and it binds on the surface.
Mm-hmm.
And so something like that, if it binds on the surface of catalyst, no other molecule can bind.
Sure. But we're going to set this up just at the level of reactions, not appealing to specific chemistry. And it's not hard, since we can write down whatever set of reactions we want. So first of all, keep in the back of your head that to get 0.3, 0.4, and 0.3, I'm going to initialize the relative quantities of E1, E2, and E3 to, let's say, be 30, 40, and 30. It doesn't matter what. It could be 30 nanomolars. It could be, but it's the relative ratio that matters. But the relative ratio of E1 and E2 and E3 is going to determine what happens next. So there's going to be a first reaction that happens. Now, there's more reactions in the system. So here's the next set of reactions. The next set of reactions is making many babies of yourself. I have to come up with a better metaphor here. But so if a given soldier D1 is the first soldier to step forward, then what does he do? With a much higher rate than the previous set of reactions, there is a catalytic reaction that makes many copies of D1. So D1 produces 2D1, 2D1, 2D1. We'll consume the E1 in the process, but we'll make lots of copies of D1. That's if D1 is the first soldier to step forward. If D2 is the first soldier to step forward, then we get many copies of D2. If D3 is the first to step forward, we get many copies of D3. That's the catalytic part. And notice the reaction rate is much higher. So whoever steps forward first, then that reaction is going to take off. That's not enough, because if a reaction takes off, well, then another soldier can step forward, and that reaction can take off, and it will. But if we also set up another set of reactions here, which are the, so I need better names here. Am I missing? Shoot. I think I'm missing, because of the PowerPoint slides, I'm missing the plus. So this is D1 plus E2, D1 plus E3. Sorry about that. I thought these slides were clean. I looked at them. Okay, but this set of reactions is the shoot your friends set of reactions. So that if the soldier steps forward, we get molecules of D1, and because of the catalytic reaction, we start to get lots of molecules of D1 quickly. Then all of those molecules clobber the other E molecules. So D1 clobbers E2. D1 clobbers E3. So if we start getting D1, it means we quickly start eating away at our supply of E2 and E3. And again, these reactions are fast enough that we start eating away at E2 and E3 quickly enough so that the chances of E2 or E3 ever stepping forward is going to be small. So likely the one soldier will be the only soldier who has ever stepped forward. This is making sense? It's simple enough. And this in itself would be, I think, enough. But through simulations, we want a really strong response. We're going to introduce another layer. And this other layer is kind of an error control mechanism. Suppose that soldier D1 stepped forward, started making many, many copies of himself. All of those copies started clobbering his opponents or his friends. Started clobbering them so they're falling to the point they're almost down. But suppose that it's not quick enough. One of his friends also steps forward. So you can have, you've produced a lot of D1, but suddenly you get a little bit of D2. Well, then you set up a reaction here, and again, I need pluses. You set up a reaction that quickly clobbers the error. But why? Like, I mean, here, if I set a reaction D1 plus D2, why wouldn't it also clobber D1? It does. But at this point, you presumably have a lot more D1 than D2. So you've sacrificed one of your soldiers to go and kill one of the other soldiers. And this, again, has a very high rate. So do all of you buy that if I set up this set of reactions where I initialize E1 and E2 and E3 in the ratio x to y to z, then in the end, I should either have D1, call it green, or D2, call it blue, or D3, call it red, and I should only have green, red, or blue. And the probability of these outcomes should simply be the ratios. I'm missing the plus signs here. So comments on this. There's a little bit more to the story here. But why? Is there any conceptual flaw here? Like, I mean, we should be able to set this up so that with a very simple set of reactions, when we do it, if we did it in a test tube, we'll get a probabilistic response. If we do the experiment 100 times, 30 times we should get green, 30 times we should get blue, 30, 40, 30 for the different types. And not only that, by changing the ratio, we can change the probability of the outcomes. So what's your name in between Seamus and Jesse? Pardon me?
Heather.
Heather. So Heather, do you see a problem with this? And why haven't people done experiments like this? Like, I mean, question. Well, I guess it's vague, but I proposed this result, and I've discussed it with chemists. And my first thought is, well, let's find simple chemistry, let's say protein-protein chemistry, where the result is fluorescent proteins of different colors. And let's set it up, very simple set of reactions, so that we put it all in a test tube, shake up the test tube. We either get green fluorescent protein, and it's dumb because so people refer to this as GFP, green fluorescent protein. But then now they have fluorescent proteins of other colors, but it's such a common acronym, GFP, that I've heard it referred to as blue GFP or red GFP. But so why can't we just do this experiment? Any ideas?
If you go back to your reactions, there's things don't just disappear or annihilate. So that could be one issue.
Right. But by the annihilation here, it would simply be a reaction that produces a waste product that's stable. So one issue would be here, I'm ignoring all the reverse reactions.
Right.
And so I think the larger issue is if I went up to, let's say, an organic chemist and I said, the problem is you can't find reactions that exactly fit this template. There are individual reactions so catalytic reaction, you can find that template, but the problem is if you're just asked to find chemistry that matches your template, In general, how do you begin doing it? And I guess the whole premise here, and the reason why somebody from chemistry or molecular biology might find this whole discussion strange, is I start off by pulling reactions out of a hat and listing them, saying this is what I want. And that entire view seems strange, because normally you start with chemistry and you describe what's going on. So that's perhaps why there's no obvious way to implement this in a test tube, even though I'd really like to see if there is. But when it comes to synthetic biology and programming DNA, I tried to explain to you, I think in some of the early lectures, that we can actually design DNA strand displacement reactions that fit any template that we want. It's a fully programmable template. And so I think with DNA strand displacement reactions, we can really get this going. And I'd actually like to do that experiment. We're going to set it up with DNA strand displacement reactions that produce, you can have DNA reactions that eventually lead to a product, which is just a DNA strand that is tagged with a fluorescent protein. And I think in a test tube, we should be able to get this experiment going with DNA strand displacement reactions such that it either we select the ratios of the inputs and we perform the experiment. It either glows red or glows green or glows blue. Does it, if we do 100 trials, in approximately the ratio that we selected, change the ratio of the inputs, do the experiment again, and kind of show that we really have intrinsic randomness here. But when I've tried to describe it to people who do the experiments and setup, they haven't pointed out why they're uncomfortable with this, but they kind of don't believe that it's going to work. And that's your reaction too, Jesse? That's somehow, I don't know, it's just not going to work. Because, again, perhaps all your bias for the people who do experimental work is that the more careful you are in controlling exactly what you do, the more you can replicate the results. And the entire scientific method that we've been taught is that science is something that we can replicate. And kind of the idea of doing an actual experiment and getting random outcomes makes people uncomfortable, maybe or not. Yeah, so Bishwash.
We're also ignoring diffusion in this, so that might also be an issue.
But I'm just assuming everything's well stirred. So,
it's really hard to get that in experimental setup.
But why? So talk to me a bit about that. So meaning, I see. So what you're suggesting is the first soldier who steps forward, or in his local vicinity, he's going to be shooting all his friends.
Yeah,
but go over to France and there's a different war going on.
Yeah.
Okay. But the thing is, the catalytic effect and the inhibition effect can be strong. So that you're over here, you're starting to win your campaign. And even though a little skirmish is starting over there, pretty soon you're just going to overwhelm it. Why won't that happen? You know, like, I mean, this is basically a winner takes all. And the winner takes all should be a random event based upon the initial conditions and not based upon, the factors that you can't control, but just a random event that happens locally. And Rowan.
So another thing is, like, I guess from the way you stated the problem is like D1, D2, and D3 are your outputs that you like specify before, and yet you're trying to create reactions with those outputs, which might not necessarily happen.
I'm not sure I understand. So
like, I mean, in the beginning you said like,
yeah,
we want bacteria to produce A, B, and C.
Yeah.
Which, I mean, in a real life setting, I assume that you would know what A, B, and C are before.
Yeah.
But
Yeah.
now you're trying to create reactions where like A and B annihilate each other, or produce a stable product. Which,
right.
I mean, might not necessarily happen because I feel like you choose the outputs first.
Sure, but I haven't, so I should have continued because, so the rest of it I don't find it interesting, but let me expand upon this. So your question is a very good one. Here, I want products A, B, and C, and I'm using the soldiers as my products. And it could be that the chemistry that involves the soldiers is not the product that I want. But so there was just one more sequence of reactions that I had here to basically address that concern. And more broadly, I stated at the outset that I wanted a fixed amount of green, or a fixed amount of blue, or a fixed amount of red. I wanted fixed amounts. But here, it seems that the amount that I'm going to get is based upon this runaway catalytic mutual exclusion reaction. But suppose that D1, D2, and D3 are not my products. They're simply intermediate types. They're simply the signal that tells me which product I want. Then I can set up another set of reactions. So here, this is really unfortunate with the messed up fonts. Damn it. Can you guys read this? So the diamond is for J equals I, and I'm missing a couple of plus signs. Okay. But here, the first, so this is just a generalization with subscripts of what's going on. The initializing reactions are the reactions where we get E1, E2, and E3 in the right ratio to produce that initial soldier stepping forward. The reinforcing reactions are the catalytic reactions, that for each soldier that step forwards, producing many copies of himself. The stabilizing reaction is the kill your friend reaction. The purifying reaction is kill each other, so that only the winner remains. This is the new reaction that I'm implementing here that addresses Rowan's concern. So this will be a reaction that, based upon a winning type, D, let's call it D1, we produce the actual output that we want, which we'll call O. So O is the actual output that we want. So green, red, and blue is O here. And we produce O from a food source, or a supply, F. And so we supply, at the outset, some reference quantity of F. And that's going to be how much we want in our green puff, if we're going to get a green puff. So the idea is, if we get D1, we'll only get D1. And then D1 will participate in a reaction that translates food into the outcome. But the limitation here is how much food we provide. So the amount of F we provide will determine the amount of the output we provide. Does that make sense, Ron? Yeah? Okay. So that's just how, if we want a specific quantity of an output, where the probability of getting that specific quantity is a function of the ratios, how we can achieve getting that specific quantity. We simply pick a winner, and then the winner, based upon a food source, produces a specific quantity of the outcome. Okay. So here, of course, perhaps the limitation with a lot of chemistry is going to be the rates. We definitely want the, and I should really call them catalyzing and inhibiting reactions. The catalyzing and inhibiting reactions to be much faster, much faster. So the diffusion concerns, they have to be overcome by rampant reactions. So whoever starts to win a little bit definitely wins. That might be hard to achieve. So that might be what the obstacle is. Okay. Now, I'll ask you guys to do this calculation because it definitely seems simple, but it's not quite as simple as it seems. Because if we want to obtain a particular outcome with a probability, how do we select the ratio of the inputs? And in particular, suppose that the rates that I have for the reactions, I just call them all 1. Suppose that the rates of those initializing reactions are not necessarily something I can control so that they're all identical. Because, of course, the chemistry for E1 going to D1 may be different than the chemistry that goes from E2 to D2. So I'll ask you guys to do this as a calculation to figure out how, if you have the rates, the K1, K2, and K3, that are given, how do you control the ratios of the initial reactions to get the probability distribution you want. But this is, in essence, just a calculation detail here. Okay. So here, I can just show you some graphs. And these graphs perhaps aren't so interesting. But here, this is just describing how well the method works through simulation. And it's through simulation of DNA-level reactions, so probably a pretty good simulation. But the simulation parameter is, if you think of it, this entire scheme, its accuracy, or how well it works, is going to be entirely dependent upon how fast the catalyzing and inhibiting reactions are. The faster they are, the better the scheme is going to work. And so let's call the ratio of the rates lambda. So in the slides that I had 1, and then I had 1,000, and then I had 1,000,000, I think, for the rates. Let's call that ratio lambda. So the ratio gamma. Gamma or lambda? What I have here? Gamma. So let's call it gamma. So let's call gamma, let's say 1,000. then 1,000 would mean that I go from 1 to 1,000, from 1,000 to 1,000,000. So here, what we've plotted is the error that we get. And the error that we get, it's an interesting question here, because even in simulation, it's not exactly clear how we should measure the error. And that's a whole other discussion. Let me give you my thoughts on that in a moment. But here, as a function of the rate separation, we plot the error that we get in these different systems. And you can see that it drops, and it drops exponentially. But depending upon how satisfied we are with the results, the rate separation issue is a real issue here, that you need much, much faster reactions to get things going. But let me ask you this question. Let's see if I can formulate this in my head. How do we estimate the error? Because, in essence, the scheme that I've suggested, in a probabilistic sense, an argument could be made that it should be error-free. Like, I mean, can any of you, to play devil's advocate, can any of you suggest how in a system we can actually get an error if we let the system run?
In this system or in a different system?
In the system. The error is hard to describe because everything's happening probabilistically. And
Maybe the amount of final quantities you get because of that mutual killing out, that might vary.
But yeah, absolutely. But the problem is that in a hypothetical, academic, theoretical world, we would simply, in terms of the experiments, perform the experiments over and over again. I said we should do it 100 times and look to see if we get 30%, 40%, and 30%. That's if we do it 100 times. But then what if we do it 1,000 times? What if we do the experiment over 1,000,000 times? The more times we do the experiment, in a theoretical sense, the closer we will get to the actual ratios of 30, 40 to 30 in the outcomes. And so how can anything go wrong? Like, I mean, things can go wrong in one trial. But if I do enough trials, I should get the right distribution. So I don't know where you guys are going in your thinking with this, but my thinking is, okay, then what we actually have to measure is the variance in what we get. Because a good system, of course, a system where you repeat the number of trials to infinity is always going to give you the exact expected value. But it's the variance we're interested in. If you do it a few times, do you get a large variance? But the variance is hard for me to wrap my head around here. It's the variance of what? So any thoughts on this? Maybe only a little bit of time to think about this. But when I tried to think about this, I couldn't properly wrap my head around, in simulation here, what we're even describing. This variance of what? So here what we've plotted is simply the percent of trajectories where we get the wrong choice in the final outcome relative to the first reaction that happened. So if the first reaction is a soldier who steps forward, what is the chance that some other outcome wins instead? But that's kind of, that's really a constrained view here, because who's to say that the overall statistics really depend upon who is the first one. What you want is the overall correct results. So anyways, I don't know that I really butted my head against this, and perhaps the fact that I'm struggling to understand what the variance is or how to measure this theoretically, maybe it points to what is an underlying flaw in terms of the experimental setup. So I don't know if this is making sense. Does anyone have any thoughts on it on the spot? Or perhaps think about it. And I'll try to formulate a homework problem that will address this. But any thoughts on this?
Maybe you can have different variants for the different outputs, or one for red, one for green, one for blue.
Mm-hmm. But how do you measure that variance in terms of studying the simulation?
So if we do it for, say, 10,000 times, I'm supposed to get 40% of blue.
Mm-hmm.
And then I run the simulation for 10 times. And
Yeah.
I'm getting 5 or times. So there's a
Yeah.
I should have got 4.
Right? Perhaps it's my ignorance with Monte Carlo methods. So the problem is, if you run it 10,000 times, what if you run it 100,000 times? Your variance starts to shrink. But that's not what we're trying to measure here, because obviously, so a system in which you had 1, 10, and 100 for your rate separation, so you had a separation of 10, is going to perform very poorly. That's my intuition, compared to one in which you have rate separations of 1,000. But I don't quite know how to capture or quantify this just through the repeated trials and statistics of it. But it's probably because I really don't understand how to measure variance with Monte Carlo methods. I know how to measure the mean with Monte Carlo methods, just do it, correct the fraction, but I don't know how to capture the variance. Maybe some of you know better than me. Or, I'll try to formulate this for a homework problem. But for this, let me return to the topic of intrinsic versus extrinsic noise and talk to you a little bit, not about this artificial system that I've presented, but about the real system, the lambda phage. I'll actually show you the reactions for it and discuss the debate which I heard raging. I heard raging in papers that were published, but also at conferences where people were nasty to each other, claiming, no, you haven't shown intrinsic noise, you've shown extrinsic noise. Okay, so let me switch. Yes.
So,
Chopin.
Interested in producing a drug, right? So, why do you want 3 different types of I mean, why just not put one ingredient and let it
Uh-huh.
go there and then release
Sure.
it?
This is just a generalization, but you could imagine that your outcome could be produce D1 versus D2 only. And then your food source could be produce your output as a function of D1 and do nothing with D2. So this way you would either get green or not get green with the probability. Was that your question? Yeah.
So, like, why do you want to get green only a few number of times and not the rest?
Oh, so the hypothetical motivation here, or the conceptual motivation, would be you want to control the amount of drug that's delivered, and so you want to control the probability of whether a given bacteria produces a puff or doesn't produce a puff. And because everything is equal and equivalent, you can't control the quantity of individual. You can't turn the dial and say, each bacteria produce a little bit more. That's hard. Because here, as you notice, we don't have a measured response. We do have an all or nothing response. And it's very hard to get an exact measured response. Make sense? Yeah. So first, I'll show you the image. And this is... I found this intriguing because when I saw this before I understood anything about the topic. And this looked like a circuit diagram. And it looks like a circuit diagram in many ways. So we've got switches, and what else do we have? We've got wires. But then a lot of it starts to look foreign. So this is an image of a genetic regulatory network in the virus. So, and some people in the field call these genetic circuits. But what this is a description, a cartoon description, just like for electrical engineers we use cartoon descriptions of wires and gates and transistors. It's a cartoon description of what's going on. It's a cartoon description of what are chemical reactions that are firing. But the cartoon description is helpful to somebody who's a real expert in the field. The key point is that what's going on is this circuitry, which is the gene network in the virus, is the circuitry that tries to decide whether the virus goes into lysis versus lysogeny. So let me write down those words. I've used hijack versus stealth, but we may as well use the real words. So lysis, So lysis is, I think, Greek, and it means to cut. So the lysis mode is when the virus decides to kill the host bacterium that it infects and make many copies of itself. The bacterium bursts open and the virus is released. That's lysis. And this is a choice And the circuitry is the circuitry that determines the choice. It's a choice that the virus makes to do. It can say, okay, make many copies of myself, and in the process, kill the bacterium. Another choice that it can make is lysogeny. Lysogeny is basically splice my genetic material into the genome of the host. So take my DNA, or is it RNA? My DNA, and splice it into the DNA of the host bacterium. And by splicing it, it means basically, I think the metaphor is apt, like you splice pieces of twine. Basically, you insert a stretch of DNA into the host DNA. But the effect of lysogeny is you don't harm the bacterium. The bacterium does what it does. But when the bacterium divides, then your information divides along with it. And this can happen for many generations where your genetic information divides with it. Until at some later time, you decide to switch back to lysis mode, where you start making many copies of yourself and you kill the host bacterium. So interesting and two distinct choices that are made. What I find interesting here is we use choice, and I don't think anybody's thrown by the metaphor, but there's no brain at work here that's making a choice. The choice is what the chemistry dictates. And that's what this circuit is trying to show, or to somebody who's knowledgeable what it shows. The key point is the switch. So there's a switch that's thrown between choice 1 or choice 2. Now the interesting part is the fraction of time that this switch is thrown. So, to perhaps explain or confuse, let me go, I don't understand at all the details of this. I only understand the outcome. And in fact, when I was trying to understand this, it was frustrating because if you read the theory about this, and maybe this is true for all of engineering, somebody has designed a clever circuit. You see the circuit design, and then they kind of describe qualitatively how they arrived at that decision. But the only way to understand it is either follow the mental steps that went into the design, or else begin to understand it by looking at how it behaves. So, looking at how it behaves, I got a hold of the set of reactions that it describes. So let me go to that. Can you guys see this is ugly? Okay, it's still kind of too small, isn't it?
Press Apple Plus and make it work.
That's the green.
I don't think I can with my tools.
With Magnum, yeah, you can.
Pardon me?
Yeah, you can.
With MacVim?
Yeah.
Not working. Anyways.
I mind you can.
But in any event, so blowing up the details here really won't help you. I just wanted to show you the format in which the data is presented. And this is a format I've massaged into my own format. But this is just a set of chemical reactions on a set of types. So instead of A, B, and C, in all the examples I've shown you before, we've had A, B, and C made-up types. Here, these are names. These are names to the types of molecules that the biochemists have studied. But that's all they are. And the format of the reaction is simply we have a type and a coefficient, a type and a coefficient, then a colon, a type and a coefficient, a type and a coefficient, and then a colon, and then a rate. So the top one, if we have something, I'll just write it like this. Suppose we had A1, B1, colon, C2, colon, 3. This would be the reaction A plus B goes to 2C at a rate of 3. So it's just a set of reactions like this. There's 145 reactions in about 70 types. So that's the size of the model. And that's the model that I think it took people quite a while to develop that describes the set of reactions that were depicted in the cartoon circuit that I showed you. So that's the set of reactions. Now here's the interesting part. And of course somebody else compiled this model for me, but reading through it, there was the discussion that the decision between lysis and lysogeny is one that should happen probabilistically, or the people who are on the intrinsic side of the debate. They say intrinsically there should be a probabilistic decision between lysis and lysogeny. And that probabilistic decision is based upon the input concentrations that you provide to the system. And so, I don't know if I have. Here is a set of input concentrations to the system. And input concentrations here, and one of the reasons why I'm showing you this format, even though it's too small to read, is in the next homework you'll be working with this file and you'll be working with running some simulations on it. But anyways, this format simply gives you a long list of the quantities. So A, B, C, 3, 5, 7. So those are the initial quantities of all these types. So we've got the initial quantities, we've got the set of chemical reactions. What can we do? Well, we can write a little simulator. That little simulator just simulates what we were discussing 3 lectures ago, which is how to track the evolution of the system. The simulator, and I'll give you my source code that does it, is simply source code that computes the probability of each reaction firing according to the calculation of the alphas over the sum of the alphas. Picks one of those at random, fires that reaction, changes the quantities, recomputes the probabilities, and keeps going. Now, the interesting part is that the biochemists have a criterion to decide whether lysis has happened or whether lysogeny has happened. The criteria is you guys can't read it, but in the first reaction, in terms of the specification here, I include something like greater than or equal to 145. And the other one is greater than or equal to 55. So for 2 specific types, they're called CI2 and CRO2. For these specific types, if CI2 goes above 145, then we can reliably say that we've entered lysis. If CRO2 goes above 55, we can reliably say that we've entered lysogeny. So these are basically, recall that in the discussion of how we simulate chemical reactions, there was the idea that we start somewhere and then we follow some trajectory through the state space. Here, we're starting somewhere, so we're starting in some common place, and we're following some trajectory through the state space. And suppose that we arrive here. This quadrant is where we have CI2 greater than 145. So we have lysis. This trajectory that lands over here is one in which we have CRO2 greater than 55, in which case we have lysogeny. And as it turns out, when we run the simulations, we pretty reliably only get one versus the other. So we never end up with CI2 greater than 145 when CRO2 is greater than 155. Or, in any event, we stop the simulation when one or the other happens. So we simply stop it when one or the other has happened. But here's the interesting part. I can take this set of reactions, I can take these initial quantities, I can take these, I call them stopping criteria, and I can just do the simulation that I was describing. And then I can count the number of times, the number of trajectories that land here, or the number of trajectories that land here. And then from there, I can estimate the probability of lysis happening versus lysogeny happening, just the fraction of times I land here versus here. This is going to be a number. And the number for the values that I've set up here is going to be 0.8. So 80% of the time I do lysis, 20% of the time I do lysogeny. And this is exactly the behavior that we're after. Because what we want is a description of the probability of lysis versus lysogeny as a function of the initial conditions. And anyways, but to continue, and you'll do this yourself in the homework, if we change those initial quantities, so we change the relative rates of, and they're not all 0. Many of these are 0, but some of them are not. But if we change some of the initial quantities of the types, then this probability changes. And so we can plot the probability of lysis versus lysogeny as a function of the initial concentrations that we supply. So recall the discussion that choosing lysis versus lysogeny, that choice might depend upon some factors. So one factor is how much glucose we have in the environment. If we have lots of glucose in the environment, then the host bacterium is probably going to thrive. Life is great. So you might want to choose lysogeny. So with a higher concentration of glucose, lysogeny might be the preferred choice. So we should see a high probability of lysogeny. And there are other such factors that dictate whether we should choose lysis or lysogeny. And the interesting part is that there are many different ways to view this. But the fact that things are being chosen randomly, it actually makes sense from an evolutionary perspective. It makes sense that if you're a given virus, you shouldn't decide all the time to do lysis, given conditions. Or you shouldn't decide all the time to do lysogeny. Instead, if you're like a savvy investor in the stock market, based upon conditions, the optimal choice in terms of your success in evolutionary terms is to have a spread in your portfolio. And the spread in your portfolio is determined upon the conditions. So if the conditions are roughly equally favorable for lysis versus lysogeny, it should be 50-50. But if they're slightly more favorable for lysogeny versus lysis, you shouldn't go 100% in for lysogeny and none for lysis. So the stock portfolio model seems to be one that's plausible in evolutionary terms, and it's one that the chemistry seems to indicate, and it's one that we can just simulate. So there seems to be a convergence of evidence that this is an accurate way of modeling things. And in any event, this was my entry into the world of discrete chemical kinetics, trying to understand what's going on with that circuit that was the switch, getting a hold of the model, and then simulating it, and seeing this probabilistic response happening, and that tracking a description of what's supposed to happen. I'm not saying this is my research contribution. This was just my way to understand what was going on. And I think that this is an example how, in nature, synthesizing probabilities was useful. And I'm trying to show you a specific construct. But here's the initial question that I had. Lysis versus lysogeny seems to be a simple function of the initial concentrations that we supply. Why is the circuit so complicated? Why 145 reactions? So my initial question to myself was, okay, let's characterize how it behaves. There's 145 reactions with 70 types. That's a lot. Can I just sit down and design a much simpler system that achieves the same function? And that, as it turns out, is relatively easy to do. You can build a system that produces a much cleaner, much crisper probabilistic choice. In other words, that exactly matches this function, but that has far fewer reactions and does so more robustly. Now, I don't think that's saying anything about nature and evolution because a number of reasons. Evolution may have followed its own path to set up this switch. Furthermore, the gene network that's involved may have other functions than simply this decision. But still, I think it's interesting to this isn't even reverse engineering. It's it's designing your own black box implementation of an input-output specification. And I think that's an interesting exercise. Okay, let's stop there. By the way, for the homework, feel free to bring it on paper Friday or email it. But you should email it. You can email it to me, but email it instead to the TA, Josh. I think his email is on the website. So you can email it, your choice. Both are equally convenient.
For this first one, the question here is how is it graded? Because I went through it, but at the end you kind of proved it was correct. This for the first problem.
Yeah.
And it's not correct, but I went through all the work. So is it worth going through and figuring out what's wrong and fixing it, or is it not the correct result? Making sure it's
Sort of. But so for many of them, I'm not interested in a specific value. But here, because it'll actually be hard for the TA to figure out, it's hard for you to figure out what your mistake is. It's going to be 10 times harder for him to figure it out. So it might be hard for him to figure out if there's a mistake in understand what's going on, or if it's just a mistake. If it's just a mistake, then I'm happy to give you, numerical calculations. I'm happy to give you full points. But it might be hard for the TA.
It's hard to figure out what.
So perhaps, perhaps try to explain what you've done at every step, so the PA can say, okay, that's correct. There must be a mistake here.
And then for problems that I would do programmatically, should I include the program?
Yeah, I should say, just include it. Like, I mean, we're very likely not going to scrutinize or run your code, but it can still be interesting.
And then, so for the second problem, I was originally doing it by hand.
Yeah.
And I was just doing individual C, individual S.
Yeah.
But when it's an overall probability, I don't see an easy way you can combine those probabilities not because they're not independent.
Yeah, like, I mean, you have to combine them at the level of individual conditions, which is, I think, you have to...
Okay, so I was then just going to write a program to iterate over all the combinations.
Yeah.
Okay.
For the second problem, the probability failure.
About graduating. Sure. Oh, yeah. But that was only because we were trying to be more sophisticated and have to figure out what the coverage is. So now I'm just making my matrix by default. I'm doing it until I get a right one. Sorry. Because you can't. Where that problem hasn't been solved yet. I think I just am not understanding. Making a matrix that the rows and columns add up to a specific value, which is what you need to solve the problem.
So I feel like that when you get to that really A family, it's not like that.
For it.
It's based off our implementation of how we define neighbors. Because
In matrix.
we were just creating a matrix where then by a matrix the diagonal is correct, and then one first indicates those two being zeros to the correct neighbors. Okay.
But regardless if it's implemented as a matrix or as objects in which you have some property, it's still mathematically defined as not solved.
Or we can have, if you get like, this one is correct, this one failing is not correct.
So, like, imagine if you had, say you had 3 by 3 groups, and you wanted every row and column to add up to 1. Actually, you wanted 4 by 4, every column and row adds up to 2. Well, imagine you could start plotting, and you want to use a random number generator to make this matrix. As you start plotting the points in there, you end up with a row that's 0 and a column that's 0, but everything else is filled in. Well, now you can't solve it. And so, if you're making bidirectional paths between your